Thanks very much, Charlie. It's my pleasure to introduce our speakers, Mike Worobey and his student, Lorena Malpica Serrano. I've known Mike for many years. He began life growing up in a small town called Salmon Arm in the interior of British Columbia, where he jumped out of, it was either airplanes or helicopters or both. Probably helicopters, to put out fires. And I think that has been somewhat of a metaphor for his scientific career since then. Mike did his undergrad at Simon Fraser University. His first project was the study of phylogenetics of galls and gall insects in my lab. He went on to much better things at the University of Oxford to do a DPhil. And then he began his faculty position at University of Arizona in 2003, where he is now professor and head of department. It's all yours, Mike.
Well, thank you very much. It's good to be here, and thank you for that nice introduction, Bernie. Thank you, Meredith and Charlie. And I'm looking through the people who are tuned in here. I see Jeff Joy, Patrick Abbott. Lots of old and great friends, and it makes me realize I should be tuning into this all the time. So I'm going to be coming to future events, but I'm pleased to be talking in this one, and pleased to be talking with my PhD student, Lorena Malpica. And the two of us, along with a whole bunch of other colleagues, have tried to really dig in and answer the question of where this pandemic came from. So I'm just going to dive right in here. Next slide, please.
So hopefully I'm trying to get it to where you have control. Are you able to advance it?
Okay, so would that be using my arrows?
I think so. Let's see.
Okay, I'm just getting click to start the remote control of the shared screen. Hey, look at that. Maybe it worked. Did it work?
Yeah, it looks like you're advancing now. Perfect.
Okay, but it looks like I can't. Sorry for the delay, everyone. I can't actually do anything with the arrows. I think when I click, it goes. Oh, wait a minute, maybe down here. Okay, I think I've got it. All right. So let me just open with mentioning, in addition to Lorena, some of the other early career researchers who were really important parts of this work. So Josh Levy in Christian Anderson's lab at Scripps, Jonathan Picar in Joel Wertheim's lab at UCSD. And so Joel was my PhD student, and Jonathan is his PhD student. So that's kind of cool. Alex Christoph, who's now at Hopkins, absolutely amazing sleuth and just general quantitative genius. Stephen Goldstein, fantastic virologist, and the rest of the crew, and actually this is actually only from one of the two papers I'm going to talk about, so there's others that I'm missing, but I'll mention. Can you guys see my cursor when I shine it on Eddie Holmes?
Yeah.
Yes. Okay, so Eddie was my de facto supervisor, so we've got 4 generations down from Eddie to Jonathan Picar, which I also think is kind of cool. And Christian Anderson was the kind of co-senior author on the Huanan paper. Joel Wertheim is so Joel, me, Jonathan, Mark Suchard are the senior authors on the genomics paper that I'm going to talk about. Okay, so wasting time. Let's not wasting time, but let's get into it. So people will remember probably early on when we first started hearing about this outbreak, that there was talk of a lot of the cases being linked to this Huanan wholesale seafood market. So here's a picture of that market with people blurred out. It's a vast market with all sorts of different stalls. And the name seafood market is really a misnomer because it turns out it sold all sorts of other things, including live mammals. Here's a shot that Eddie Holmes actually took in one of the stalls at that market. This is a raccoon dog. It was 2014, October of 2014, when Eddie was taken to this market when he was visiting collaborators in Wuhan. He was setting up collaborations to look into emerging infectious disease. And people took him there and said, this is a place where animals with potential pathogens that could jump into humans are housed. And so this is a shot of one of these animals over one of these open drainage grates in a nice little green cage. Oh, haywire, went haywire. And here are other pictures taken now in 2019, December of 2019, with a bunch of other animals that were there. And part of what we did in our paper was establish what live animals were sold there, building on absolutely brilliant work by Xiao Xiao and Xiaomin Zhou, who did this unbelievable study from 2017 through 2019, where Xiao Xiao went every month to this market and 3 other markets in Wuhan that consistently sold live wildlife. And he asked the vendors, what illegal wildlife are you selling this month, and how many raccoon dogs and civets and whatnot have you sold? So meanwhile, the WHO did a big joint study with China. And so in early 2021, they put out a report where the Chinese delegation in that study basically said, yeah, no live wildlife was sold at that market. So doesn't seem like that's how the pandemic started. And then a few weeks later, out of the blue, Xiao Xiao and Xiaomin Zhou and other colleagues show month by month what was being sold in these markets in Wuhan. But what they weren't able to show in that paper, or didn't show, was specifically at the Huanan market, what was being sold in November and December of 2019, which are the crucial dates. So we were able to show, yes, they were selling raccoon dogs, red foxes. This is a photo of a red fox, not from this study, but from another study. Lots of animals that could have harbored the progenitor of SARS-CoV-2. Okay, so for the last year and a half, I have been really trying to falsify the hypothesis that this outbreak and now pandemic started at the Huanan market. And the first stop on that was to challenge the notion that the Huanan market really was associated with a preponderance or a plurality of the early cases in Wuhan. And so one possibility is, okay, it's a live wildlife market. It is the kind of place that was associated with the original SARS 20 years before. So maybe it's just a case of ascertainment bias, where maybe they were looking for cases of new emerging infectious diseases at wet markets, and that's why these cases were ascertained there. Meanwhile, all across Wuhan early on, there were thousands of other cases that just weren't ascertained. That's a possibility. So to cut a long story short, if you want to look at my perspective from November of 2021, when you peel back the layers of the onion from articles published in The Lancet to notifications put out by public health authorities in Wuhan and Hubei province, all of which could have been impacted by ascertainment bias if people were looking more at the market than other places. You get back to this person here, Dr. Zhang Jixian, and she basically discovered the outbreak in her hospital on December 27th. She saw CT scans of 2 patients that were husband and wife. She immediately thought it looks like SARS. She realized that they were in the same family, so it was probably infectious, human to human transmissible. She also forced their son, adult son, who was healthy, to have a CT scan, and his lungs were full of lesions as well. So basically on December 27th, she had worked out there was probably a new SARS-like pathogen, human to human transmissible, and you could be asymptomatic, but still very much carrying this virus. None of those cases were linked to the Huanan market. So the outbreak was discovered without a link to the market. But then the next 4 cases that popped into her hospital over the next, let's see, 27, 28, 29, up to December 29th, all were workers at the Huanan market. And so she and the other people at her hospital sort of rang the alarm. They contacted Wuhan CDC and Hubei CDC and said, look, there's a new outbreak. A bunch of cases are linked to the market. But what's important is they weren't looking, they weren't searching in any way that would be focused on the market, yet more than half of the early cases were from the market. And so of the early cases, there really were a bunch, a huge number linked to just this one market the size of a Home Depot in a city of 11 million people. All right, I think I really need to pick up the pace here. These are a couple other papers, one of with Jeff Joy that we published along the way that I'll return to. These are just sort of important because they help build up the methodology that we used for the genomic epidemiology paper that I'm going to end on. So, but mostly I'm going to talk about this paper on primarily spatial data and data within the market. Okay, so we used a whole bunch of different information sources, from epidemiological to genomic to where the people who are the earliest known COVID patients in Wuhan lived. Okay, so the Chinese authorities went through the records of people who had COVID with onset in December. There are 174 of them. And they actually interviewed them and got the home address for each of them. So they had the latitude and longitude of where these people lived in Wuhan. And they asked questions like, did you work at the Huanan market? Did you shop there? Had you visited there? And so it's a pretty rich data set. But the way that they presented that to the world in this report of the combined WHO China study into the origins of the virus was this map of Greater Wuhan, 8,000 square kilometers. So this is a big area. And here are the 164 cases that were within Wuhan proper. There's a few that are outside of Wuhan in that 174. 164 are within Wuhan. But they mapped them with, these are the residential locations of those cases, but they mapped them with dots that were 2 kilometers in diameter. And so when you get to central Wuhan, where most of the cases are, the dots just overlap one another. They're blurry. It looks like someone's like Xeroxed this like 1,000 times to get it to be such a shitty low resolution map that you would never like share with anyone in the outside world. And yet that's what the WHO presented after a year of investigation, the world's shittiest map. And so those of us interested in this were like, you know, great, this is interesting, but it doesn't give us the actual latitude and longitudes to actually do the kinds of tests that we want to do, to ask questions about, you know, were these cases closer? Did they live closer to the market than expected? Were they more centered on the market than expected? But after a year of me and the rest of the world just kind of dismissing this as, you know, somewhat useful, but mostly useless, I realized that there was another map that was in a different part of the report that I had kind of overlooked because it didn't have any landmarks like the outline of the map of Wuhan that related to anything that I could discern. But it had much smaller dots representing cases, presumably in central Wuhan. And what I realized is you could actually overlap these smaller dots in that map with the bigger dots in the shitty map, and start to actually get a much better idea of what was going on in those the high density areas. And in fact, we were able to precisely locate 155 of the 164 cases in this way, and to use to essentially reverse engineer those maps into latitude and longitude to start doing some proper analyses. Okay, and so what do those proper analyses tell us? Oh, here's a pretty map of what the distribution of cases look like in central Wuhan. Blue cases are cases with no link to the market, no known link. They were asked and said, nope, didn't shop there, didn't work there, didn't visit. The yellow ones were people with a link to the market, almost all of them people who were actually vendors or people who worked at the market. And you can just see visually, there's, you know, quite a pattern of cases being close to the market. But what you don't know is the kind of null expectation of where cases would be likely to live, you know, if they were just sort of randomly randomly distributed in Wuhan's population. So for example, maybe this is the highest density population, highest density area where people live in Wuhan. And that's just, the cases are just reflecting that. Or it's the highest density of older folks who are more likely to show up with COVID symptoms. So you need to know the background of the population. And for that, I'm hoping I put the slide in the right place. Okay, hang on, wait a minute, it's here. Okay, so Lorena, can you just take it away now and talk about, fairly quickly talk about what we did?
Okay, So we wanted to test, like we want to build null distributions that were just reflecting the population density in the city, so as to say it really originated at the market, not because it's the most densely populated area, but because it started there. So to do that, we took raster files from world pop density that had pixel values, and the pixel values
world pop density. Just maybe explain what that is for people.
Oh yeah,
who
world pop density is a website that collects a lot of contains like it's a database of maps of population density and other geographical information, and it's stratified by age. So what we did is we took these maps, we took the pixel values that reflect the population density and matched them to the age groups in the WHO report. So we had a map of the city with the population density matching these age groups, and then from there we would sample at random 155, and see where in the city they would fall. So we would be getting a bunch of pseudo replicates of 155 cases that would be distributed in a way that reflects the population density. And we would compare this and their distance to the market to the actual cases and their distance to the market.
Awesome. Okay, great. And so with that null distribution, we're able to ask these simple questions. Are the cases from December in Wuhan closer to the market than can be expected by chance? And is there center point, if you just kind of take the center of mass of these cases, is it closer to the market than expected? The idea being, wherever in Wuhan this outbreak started, if it then sort of started disseminating into that local community as it moved into local shops where residents are shopping, and then the residents take it home to their nearby apartments and homes and infect others in the community. The idea being that it radiates out through time into the community, starting in the area where it began spreading. And this central tendency, this center point is how we look at that. And so the answer is yes and yes. That if you look at the median distance of the December cases to the Huanan market, they're 4 kilometers. But if you look at the median distance to the market for the Wuhan population, as in the way that Lorena just described, it's much higher, 16 kilometers, and so that's highly significant. And an important thing here is we looked at the possibility that that maybe there was some ascertainment bias even in the data that we had, but if there was, our results were unbelievably robust to ascertainment bias, because you could take that cluster of locations and you could then go in, and we did this, and say, okay, what if I remove the case that's closest to the market, throw it out, and now run the stats again? Are the cases still significantly closer to the market? And let's do that again and again and again. You could remove about 68% of the cases before losing significance. So this is a very robust signal. And the important thing here is, back to this map here, where you've got these blue dots, which are the cases with no link to the market, they actually lived closer to the market than cases that were linked to the market. And if you think about that, that's kind of almost all you need to know to understand that this pandemic started at the Huanan market, because there's really no way to explain a pattern where the cases of just people who are living their lives but in Wuhan, but have no contact with the Huanan market, of all the places they could live throughout the city, these early cases are very tightly clustered right around the market. These cases that have no known link. And again, you can go in and remove the ones that are closest to the market over and over again, and remove 2/3 of them before you lose significance. And I'll just, without spending too much time, say that the central tendency or the center point of the whole, let's go back to the big map of Wuhan. So of all 8,000 square kilometers in Wuhan, this little dot, if you could even discern it here, this black dot where my arrow's pointing is the center, it is the market. And the center point is less than a kilometer away from the market. Highly significant, you know, no reasonable way that the early cases could be so centered on that point by chance, if it didn't start from that area. And let me just show you another couple cool things, that if you look at the actual the highest density of where those cases early on lived, and use something called kernel density estimates. This 1% contour is essentially the highest density area that contains 1%, the top 1% of the probability mass. So in other words, given the distribution of where cases were in the city, this is the little shape that you would draw of the area that has the highest probability of having an early case associated with it. And it just so happens that the Huanan market lies within that 1% density contour, which is just insane. Like, the data we collected were just the data. I did all of this reverse engineering to get these latitudes and longitudes without any relationship to the Huanan market. And then when we crunch the numbers, this is what they indicate. Okay, so let me wrap up oh boy, I have a whole section of the fact that environmental samples. So early in January, well, basically January 1st, the market was shut down. But on that day, and then on January 12th, researchers from the China CDC went in and took swabs of surfaces across all sorts of different stalls, including stalls that we later learned sold live wildlife. And the live wildlife was sold in the southwest corner of the western section of the market. And that's where most of the positive environmental samples were sold as well. And it turns out that knowing that a stall sold live wildlife was as good or better a predictor of an environmental positive sample than knowing that there was a human case that was sampled in a stall. So very, very powerful statistical evidence that the presence of live wildlife was telling us that that surfaces in areas near where wildlife were sold were contaminated with the virus. And just to sort of go back to this green cage that I mentioned with the raccoon dogs early on, this is another shot that Eddie took in the same stall. Those are raccoon dogs, little beady eyes there. These are some birds. The raccoon dogs are stacked right on top of the birds. The stall that had the highest number of environmental positives and the ones that were most associated with animal sources was the stall that Eddie took those photos in. So photos taken in 2014, but the environmental samples sampled on January 12th, 2020. There were 5 of them, 2 from carts used to move cages, 1 from a ground surface, 1 from a metal cage, and 1 from a feather remover machine. And so we can't say that the pandemic started in this particular stall, but it wouldn't be a bad bet. And it turns out that this photo I showed you that Eddie took, that's the stall right there. Okay, so again, this photo of the market, this is the western section here. This is the corner where the wildlife was sold. This, to me, puts into perspective the fact that it's a city of 11 million people. It's bigger than New York City. Here's some cars. Here's the market. This is not a huge area. Of all the places that a pandemic could be an early outbreak could have been associated with geographically in a city this size, it's surprising that it's one of the 4 places, small places in the city that sell things like raccoon dogs and foxes. And in fact, we looked at this with social mobility data from people like posting things on the Chinese versions of Instagram or whatever when they go to a shopping mall or a seafood market or wherever. And you can just see these are visitor check-ins. And so there are some markets that have many, many times more check-ins than the Huanan Seafood Market. The Huanan Seafood Market is not the place in Wuhan where everyone's going every day. It's not a high traffic area. It's not a likely place for a pandemic to arise if it wasn't due to the market itself. And with that, I'll end quickly here just by saying the other paper with the genomic epidemiology, the punchline there is there's like more than a 99.5% probability that there were 2 or more jumps into the human population from the source of this virus. There's the early lineages called lineage A and lineage B pretty clearly had separate origins. So SARS-CoV-2 is kind of like HIV-1 and HIV-2 or HIV-1 group M and group O. They had separate, they entered the human population separately. And this is buried in the supplementary materials, but it's kind of my favorite figure. And if you can understand what this figure is showing, you can really understand the paper. So here's lineage A, here's lineage B. Each of these steps is 1 mutation. So here's in this tree, the root is a lineage A genome. And then these are also lineage A genomes that this one's 1 mutation away from the root, 2, 3 mutations away, 4, 5 mutations away from the root, and so on. Lineage A is more similar to viruses that we know circulate in bats. So the thinking is a lineage A-like virus must have been the ancestor of all SARS-CoV-2. Our paper shows that that's probably true, but it's still possible that lineage B was the ancestor. Lineage B is the only virus that had been found at the Huanan market while we were doing this work. And so that presented a challenge that if lineage B seems to have been derived from lineage A, and lineage A is the sort of bat-like virus that was probably the most recent common ancestor of the outbreak, then maybe the market was just an amplifying event of an outbreak that was already, around for a month or so, because each of these mutations takes about 2 weeks to accumulate. But what we found was a couple of things. First of all, this doesn't make sense. This phylogeny doesn't make sense with a single origin with the molecular clock. And you can kind of see it here that the lineage A genomes these are all sampled over a similar time period. So if things really evolved from lineage A, the lineage A root to tip distances should be the same as lineage B, but you can see visually lineage B is way out ahead of lineage A. It's gone many more mutations from the root than lineage A has. And just to summarize this, we show statistically that this just completely violates the molecular clock. Doesn't make sense. It makes sense if there was 2 separate origins though, because then each of these jumps is actually a separate phylogenetic tree. This is one of the roots, and this is the other root, and it doesn't matter when they occur. And in fact, it's quite likely that the lineage B jump happened first, and the lineage A jump happened later. And the fact, again, that each of these jumps or each of these lineages has a massive basal polytomy. So here's the sort of ancestor of lineage A. You have all of these lineages emanating from it with different mutations, right? All of these little branches here are different mutations. So this is called a polytomy. And same for lineage B, massive polytomy. To cut a long story short, when you simulate an epidemic starting with a single introduction into humans, you essentially never see a situation where you have 2 large clades with 2 huge basal polytomies. You only see a basal polytomy at the root. And so this has the hallmarks of being an introduction here with its basal polytomy and an introduction here with its basal polytomy. And when we're putting them together, we're actually putting together viruses that had an ancestor in perhaps a raccoon dog. And so as we were finishing this up, we were like, holy crap, it's pretty clear that lineage A probably jumped later. It's virtually certainly a separate jump than lineage B. And since the geographical information shows us so clearly that the virus emerged at the Huanan market, both of these jumps probably happened at the market within weeks of each other. And the lineage A jump having happened later on is probably the explanation for why only lineage B viruses had been sampled either from patients or environmental samples while we were doing the study. So we wrote this up and said, you know what? Lineage A has got to be at the market, just at lower lower frequency. It had to be there. And as we wrote that up, George Gao and colleagues released a bunch more information on environmental sampling. Let me say before that geographically, we also had information on lineage A genomes, because there were 2 lineage A genomes where we knew the location, and they were ridiculously close to the market. So we had strong evidence that lineage A was also connected to the market, even though it hadn't been sampled at the market. So we wrote that up and said, listen, lineage A was obviously at the market as well, just at lower frequency and probably emerged later. George Gao then put out a preprint showing, oh yeah, by the way, on a glove of one of the workers at the market, there was a lineage A genome. So it all makes sense, and no other explanation can make any sense of these data. But it's all very straightforward with the way that that SARS-CoV-1 emerged, and so many other pathogens emerged. So conclusions: both lineage A and B diffused into local neighborhoods around the market before spreading more widely in Wuhan. The market was clearly the epicenter of the pandemic. There's really no doubt when you put these 2 papers together and understand them, there's really no doubt that this is how it happened. It's as clear as many other things in science that we just don't really entertain. There's really no viable alternative hypothesis. That's where we're at with this, even though that is not disseminating very widely into the Twitter sphere or even academic circles. But we still don't know lots of things. Which animals were involved? Did they come from a farm or from the wild? If they came from a farm or the wild, was it Hubei or elsewhere? And those animals, those intermediate animals, which bat population was the source of their virus, and so on. And so with that, I just want to say that this is not, our work has been sometimes described as circumstantial evidence, the geographical evidence. It's not, it's physical evidence. The physical locations of where people who physically had COVID existed is incredibly strong evidence. We don't have evidence like that for any other emerging virus. And it's very similar to the evidence that John Snow had, where you had cholera cases in Soho in London. John Snow mapped them out. John Snow estimated that the center point of those was very near the Broad Street pump. That was enough to convince skeptics to remove the handle from the pump, and lo and behold, the cholera outbreak stopped. But it wasn't until decades later that anyone showed that this was a waterborne pathogen that was Vibrio cholera. And now we have the same situation for SARS-CoV-2. But this kind of evidence should not be dismissed as anything less than what it is, which is extremely strong and almost miraculous that we have it. And with that, I'll thank some funders and thank all of you for listening. Sorry for going on as long as I did, and hopefully we have time for discussion.
Great.
And Meredith or Charlie, do you want to moderate the questions rather than me?
Sure, we can help with that for sure. Thanks very much, Mike. That was fantastic. And Lorena, thank you very much for that presentation. We have quite a few questions in the chat. We have one person with a hand raised, Dan Janies, if you want to go ahead and ask your question. For those of you who put your questions in the chat, please raise your hand. We'd love to hear from you. And otherwise, we'll start working through those and try to get some of those questions into the discussion. Go ahead, Dan.
Thanks. May I begin? Yeah. Hi, Dr. Worobey. I'm Dan Janis from the University of North Carolina at Charlotte. And your supplemental figure 19 seems to me much more simply interpreted as if you added a bat outgroup, bats, to lineage A than lineage B. And I don't know why you reject that, because it's exactly what we saw in most of your study is exactly what we saw in SARS-CoV. There was bat to human transmission, then transmission between civets and humans in the markets. Furthermore, there's evidence of bat to human serology, bat-like viruses creating human serology in 2015 in Huanan province of China, published in 2018. So I think there are much more simpler examples for your study, and even your own supplemental data. You have bat to human transmission in many of the figures as the first events.
So you're talking about the absence of an intermediate host and direct.
Yeah, there is no in SARS-CoV and SARS-CoV-2. There's a lot of evidence for phylogenic evidence for bat to human transmission, and then later in SARS-CoV-1, SARS-CoV, there's many exchanges between humans and civets in the market, but that's not the fundamental origin of the pandemic. That's a local event that may be important to how these things get started, but that does not take away the bad fundamental origins.
Yeah, you seem to be maybe missing the point that at the Huanan market where this started, there were no bats.
Yes, the bat human transmissions probably occurred well outside of Wuhan, and we don't know where. There's some serological evidence that it was occurring in Yunnan province in 2015 , and there are hundreds of
there's serological
SARS-like viruses in the wild.
Evidence of bats, bat viruses, sarbecoviruses infecting people who lived in villages near bat caves in Yunnan, but not SARS-CoV-2.
We actually don't have the sequences, so we don't know. But
right, yeah, and so again, if you think about a market the size of Home Depot in a sea of 11 million people, there is absolutely no reason to imagine that if it had arisen in some other part of China with a bat to human origin, that of all the places in Wuhan, it would end up in the one market that sold live raccoon dogs. It's an insane coincidence. And at any rate, I don't know if have you read the genomic epidemiology paper, because
certainly,
it establishes very clearly. I mean, you can always say, well, I like the 0.5% probability that there was a single origin.
Well,
that's what you're doing
you're ignoring
right now.
The whole outgroup question. I don't want to get into your terminology, but if you're not studying the possibility of an outgroup,
there's not an outgroup. If you want to talk about terminology, outgroup is the wrong term. At any rate,
how so?
What you're doing is essentially saying, I like. Sure, you guys have shown there's a 99.5% chance that probability that there were two origins, but I'm going to go with I like the 0.5%, and so that's fine. I think we should move on to Barrett's question. Thanks.
Yeah, so I think next we actually had a raised hand from Megan Greicher. Megan, do you still have your question?
Yeah, I still have my question, but it's in the chat, and I actually really want to hear the answer to Barrett's Lenin's question. So I'm just gonna, table that.
Okay, wonderful. Barrett, you can go ahead with your question.
Great, thank you. Wonderful presentation. And given the weight of the geographic epidemiology, the statistical processes, the genomics that you have shown in this series of papers, outside of a political agenda, can you offer any reason why so many people still are pushing a lab-based release?
I think it goes beyond just a political agenda, but there are political. So let me just start by saying, I think for most people who object to it, it comes from a perfectly good place of, no political agenda, no sort of personal agenda, just it seems crazy to them that a pandemic started in a city that had the Wuhan Institute of Virology that studied bat coronaviruses. I had that same feeling of wondering. That seemed like a crazy coincidence to me too, and so I have been skeptical. I initiated this letter to science that actually really revived the lab leak theory, saying, let's study both. Let's not dismiss the lab leak theory before we have evidence. Let's look at it. But what I then learned, well, first of all, I talked about some of it. I tried to challenge it myself. Maybe the market was just ascertainment bias. Well, no, it turns out it wasn't. Our paper itself is essentially a test of the lab leak theory, because if the geographical locations of those early cases had nothing to do with the Huanan market, then you're left with, well, it wasn't the market, so maybe we should look at these alternatives. So it is a test of that. But and I haven't talked about this yet, but here's the thing. We showed in a science paper in 2021, Picard et al. 2021, where we're timing the ancestor of the outbreak in Hubei, that if you take the SARS-CoV-2 as it existed in 2019, and you drop that into some rural area, into a human being in China, 99% of the time, it will not establish itself as a successful human outbreak. Even in Wuhan, it's only like 66% of the time or 70% of the time. And that is a key point, because it means that if SARS-CoV-2 was going to emerge, it was going to emerge in a big city. And Wuhan is number 10 or 11 in terms of big cities. And so the question is, in terms of that crazy seeming coincidence, it then becomes, okay, is it that weird if the virus was going to emerge in, say, Beijing, Shanghai, Hong Kong, Nanjing, Wuhan, or a few of the other big cities? So first of all, it then becomes like more of like a 1 in 10 kind of coincidence, which is, less than the alpha that we typically use for science. It's not that weird a coincidence. But then what I have done is looked at other places. So Beijing also has a massive center that studies bats. They just put out a study, bat viruses. They just put out a preprint that I reviewed, where they've looked at like more than 10,000 bat viruses. So I guarantee you that if this virus had emerged, or something similar had emerged in Beijing, we would have precisely the same narrative, that the virus emerged from a lab leak from that guy's lab in Beijing. And there would be precisely the same amount of evidence, which is the apparent crazy coincidence that this virus emerged in a city with one of these labs. And that's That's all that there is, because when you dig deeper, as I have done, and look at, for instance, where the cases actually lived, they don't live anywhere near the Wuhan Institute of Virology. If you ask who were the people who actually showed up in the hospitals early on, they're not anyone who worked at the Wuhan Institute of Virology. The evidence, not only is there not evidence for the market origin hypothesis, there is now incredibly strong evidence against it. And so then that takes me back to your question of political agendas. Most people don't have a political agenda. They just think it's a weird coincidence. But there are certainly people with political agendas that are using this, some of them in ignorance. They really just don't understand or maybe genuinely believe that it was a lab leak. Some of them, I think, totally cynically. Some people are using it to advance their reputation. People who have for a long time argued that we need to really clamp down on virology research because it's just too unsafe. There are prominent people who are essentially amplifying the distorting the picture in terms of how likely a lab leak was as the origin of this pandemic, because it makes them look like they were right and is something that can make them more influential and make them the kind of people that get invited by Rand Paul to give testimony in Congress. So there you go. That's my answer. Any thoughts on that, Barrett?
Oh no, I agree completely, and I hope that this series of projects that you've done, as well as from other sources, really help us finally say that, you know, sometimes epidemiology works and sometimes things are coincidences. And if we chase the wrong, nothing good, and actually we're harming. So really doing the work to find where the bad guy was is what's important. And I think your work, and as I said, others have really helped to hone that down. Thank you so much.
Thank you. Appreciate that very, very much.
So we've gone over the hour, but I just wanted to give Lorena a chance to answer. She was answering some of the questions in the chat. I think we've lost Megan now, but Lorena, would you like to give any more, just to close out the session here? Do you want to give any more context to any of the answers you were giving in the chat?
Yeah, so one of the questions was that regardless of the origin, the null hypothesis could be discarded, like even if the origin was somewhere else in the city. I think I want to say no, because so if it didn't originate at the market, we would expect that the actual cases to be at a similar or bigger distance from the market as seen in the nulls. And then the other question was, why did we use age structure data? And that was because we didn't only care about the population density, but population density of the people in the age groups that were more vulnerable to COVID.
Gotcha. Okay. Okay, thank you very much. We're going to have to close down. There are a lot of great questions still in the chat. That will all be recorded, you know, in the actual recording. You'll be able to see all those. And, you know, Mike, Lorena, I hope you'll have a chance to look through that. Thank you both very much for an outstanding presentation. Thank you, Bernie, for handling the introduction. And thank you, Meredith, as always, for organizing everything.